Okay, hello. Dr. Johansson, whoops, sorry. Okay, we're going to talk just briefly about tissue tropism. And I want you to understand some of the factors that affect which cell types a virus will infect. So tissue tropism depends on a couple things. The one you're most familiar with is the idea of receptor. So a cell has to be susceptible to a virus based on it displaying the receptors that allow the virus to bind to that cell and enter. But in addition to that, the cell must be what we call permissive, which means it must have the right host factors being expressed that the virus needs to complete the virus replication cycle. And we'll talk about some examples of that in just a minute. And then the third part that influences a virus tissue tropism is the host's immune response and the virus's ability to block it. So this goes a little bit further than just straight tropism, but to have a productive infection, a virus needs to be able to make more virus and spread. And usually that involves knocking down the host immune response, especially when we're talking about multicellular organisms. So let's look at each of these factors. I like this picture because it gets you to think about the different kinds of tropism. So where this virus can go and infect. And so you can define it at the level of the host. You get a little deeper into this tissue and you can get specific to the different cell types within that tissue or cell types that are not making up an organized tissue, such as macrophages. One thing that's really important to think about is this idea of the host tropism, right? So this is really important when you're considering an animal model for making a virus. So for instance, for studying a virus. For instance, SARS-CoV-2 infects humans, even though rabbits have the ACE2 receptor, the SARS-CoV-2 does not bind to that specific receptor. And so rabbits are not a permissive, why am I saying rabbits? The example I was trying to give you was that SARS-CoV-2 infects humans by binding the ACE2 receptor. And when looking at a mouse model for SARS-CoV-2, they found that even though the mouse expresses the ACE2 receptor, the human CoV-2 virus cannot bind to the mouse ACE2 receptor. And so the mouse would not be a good model for looking at or studying SARS-CoV-2 infections. And host tropism, we'll talk about in a second too, can change. And a lot of times this is called zoonosis, where the host has changed from animal to human. Okay, so general overview of what tropism means, and what we're generally most interested is in the specific cell types, because that relates to the tissue and then can relate to the host. So just to remind you of the virus replication cycle, this first is showing you entry, and that's via receptor binding. So this is where I mentioned the cell has to be susceptible to the virus. So it has to have the receptor to even start initial infection. But once the virus gets in, remember what's happening inside the cell is that the virus is trying to replicate its genome, express its proteins, assemble, make new viruses. And so the virus relies on host proteins and the host cell machinery. So most viruses, except we think for these megaviruses, which I'll just mention briefly, don't encode their own ribosomes. So they're dependent on a actively metabolizing, translating cell that is expressing ribosomes. Lots of host proteins are required for most viruses, and we'll talk about that in just a second. And I just want you to remember that a virus is an obligate intracellular parasite, and the obligate intracellular part is it's dependent on a host cell for replication, and it's not just for the location of the host, inside the host cell, but it really requires host-specific proteins. So I just wanted to give you a feel for the different classes and remind you that there are different types of virus genomes. So from DNA to RNA, there can be single-stranded or double-stranded RNA and DNA genomes, and that the genome size varies quite a bit. That means that, or let's see, I guess what I wanted to do here, sorry, I didn't flip my page. Remember that E. coli, so that's a cell that can replicate on its own. It has all its own genes its need, has a 4.6 million base pair genome. Okay, you can see these are in the kilobase pairs, so much smaller. Humans, our genome is about 3 billion base pairs. And the largest type of virus we've isolated in this mega mimi mama viral new viral family. These have 1.1 million base pair size genomes. So there are some really strange, super large viruses, and these are the ones that some of them actually encode their own ribosomes, kind of like maybe a potential missing link between cells and the viruses that most of us are used to talking about. But other than this mega mimi mama viruses, viral genomes are very small. And what's even more important is the number of proteins. So this was just kind of an interesting range of proteins, but I put together my own spreadsheet that showed you more detail. And so I've kind of arranged it from small to large, just pulled out some viruses that you're probably familiar with. But something like rabies and hepatitis B, they only encode 5 proteins, and those are basically their structural proteins and the minimum they need for copying their genome. Other viruses you can see have more and more proteins. The big double-stranded DNA viruses, varicella zosters, the chickenpox or shingles, smallpox, which we've eradicated, and there's that big mimi mama mega virus. They're thought to encode about 900 of their own proteins, and this is one type of virus that we think can be infected by another virus. Really strange. these viruses infect other single-celled organisms, so they're not human viruses. But smallpox, cytomegalovirus, definitely human disease causing, and they encode a lot of proteins, 160 to 200, which is quite a lot more than most of the viruses we deal with. CoV-2 is interesting because if you see here, MERS and SARS, well, especially MERS, they say about 10 proteins. CoV-2, there's suggestions that there could be about 29 proteins, though I put the asterisk because we don't know if they're all expressed. So this is a really strange kind of outlier, but since it's such a new virus, we're still figuring things out. So hopefully from this chart, the point is you can see why viruses would depend upon their host cell or host factors, because they just don't encode a lot of their own proteins. They encode the bare minimum to make a virus, but not everything else that's needed for replication and assembly. Some viruses have very broad tissue tropism. So Zika virus, its receptor is something called AXL. And this is a tyrosine kinase receptor. It binds to growth factors. It's very common. So that is why Zika has a much greater tissue tropism than something like HIV, which uses a very specific receptor. So let's just look at HIV. HIV uses the CD4 receptor here, and then it also uses a co-receptor. So some HIV cells infect T cells, some HIV, did I say HIV cells? HIV viruses infect T cells, some infect monocytes, macrophages, some can infect both, right? But very, very specific. So HIV has a very small, I guess you would say, cell tropism. It doesn't infect a lot of different types of cells or tissues. SARS and SARS-CoV-2 is an interesting one. We know the main receptor is ACE2. We're learning that this virus also needs a protease. So TMPRSS2 is one of the most common ones, but there are data showing that furin, which is another very common cellular protease, can actually seem to extend the tissue tropism of SARS-CoV-2. So receptors and co-receptors can broaden or narrow the tissue tropism for viruses. And zoonosis is really a change in tropism. So it's a common salicylic acid molecule that influenza binds to, but there are different ways that salicylic acid can be attached to the receptor. So there can be an alpha 2,3 attachment or an alpha 2,6 attachment. So you want to know what that is, go look at your chemistry, but it's how that modification is attached to the receptor, and that can change its tropism. So for instance, birds use primarily the 2,3 attachment. Humans, we more in our upper respiratory have the 2,6, in our lower respiratory tract have the 2,3. So most human viruses, influenza, are using this receptor, but you can get recombination in little piggies there, and they have both receptors, and so they can make new genetic combinations and change that tropism from being something that was tropic to birds or tropic to swine to being tropic to humans. that's all focused on receptors. So remember, even if you have the receptor, doesn't mean you can have a productive infection. That's where the host factors come in, and lots of the host factors that are required are involved in translation and transcription. And especially translation, because all these virus genomes need to be translated. And RNA viruses don't ever go through a DNA cycle. So they're translated as the viral genome. And so they rely on different host factors to allow that to happen. And again, they also rely on host factors or host machinery to make a productive infection. So this is just kind of showing you some of the different proteins required. This is Ebola, with lots of colorful pictures, just to kind of help you see there's a whole bunch of virus host factors that are required for an Ebola infection to be productive and to make more virus. And this is just a partial list of some of the host factors that have been characterized and the viruses that require them. So a virus might be able to get into a host cell through the receptor, but if the host cell does not express the specific protein needed, the virus cannot go through replication and make new virions. The other part of tropism is really this interaction with the host system, and this isn't directly tropism, but it has to do with pathology, and if the virus is able to make a productive infection, because you might have lots of virus starting to be produced in the cell and then the host shuts it down. So interferon is a major cytokine that's triggered when a host cell is infected, and through that there are lots of other genes that are upregulated, and the cells communicate to each other to try to stop this virus infection. Of course, viruses are going to develop or evolve counter defenses. So this is what they're learning about CoV-2, and it has multiple defense mechanisms to stop or block the host immune response. And a lot of these are on these viral accessory proteins, like you can see these open reading frames, 9B, 9C, 3B. They haven't totally characterized what these proteins do, but one of the ways you study it is you mutate the virus and remove that protein and see what happens to the infection. And so you can kind of, that's a reverse genetics. You can kind of work backwards to see where these virus factors are affecting the host response. All right, so you've got receptors, you've got host proteins that need to be expressed, you've got a host defense response, you've got a virus counter defense response. And then one of the final things I want you to remember is this is all connected by the route of transmission, right? So the virus has to get to the right cells, right? So you can start to understand why viruses are transmitted in certain ways, because that allows them access to the tissues or cells that they can infect, right? So for example, SARS-CoV-2 is mainly a respiratory virus. So it is transmitted through the air so that it can get to your nose and lungs. But we also now know that CoV-2 can cause gastrointestinal problems. And they're finding RNA from this virus in fecal matter. So this virus may have two routes of transmission, fecal oral and respiratory. So the potential with fecal oral is if you ingested contaminated food or water, you might be susceptible to the gastrointestinal pathology of CoV-2. What was I going to say on this? So this is just kind of showing a little bit more about where you see pathology and how it's connected to transmission methods and tissue tropism, right? So how is that virus getting to where it needs to get to cause the infection? And then remember that in order to be a successful virus, the virus wants to be able to get in, replicate, and get out to a new host. So a lot of the pathology that we see, the really dangerous pathology due to a virus infection, a lot of it is due to our immune response. So yes, coughing and sneezing is a great thing for viruses that are respiratory viruses to trigger, because it's going to allow the virus to get out and hopefully find a new host. But other things like fever and cytokine storm and the chills and headaches and things that we associate with the common cold and the flu and other virus infections, those are really due to our immune response, right? So it's not due to the virus killing a few cells, it's due to our immune response to that virus infection. Okay, I think that's what I wanted to say about tissue tropism. This will connect to a lecture we'll have a little bit later, because one of the cool things is that we know that for some viruses, we can actually modify them and alter or manipulate their tissue tropism. And so this is just an image of adenovirus. Adenovirus is a virus that's been studied for a long time, another common cold causing virus, but it has a DNA genome and it's easy to manipulate and change these outside spike proteins and what they interact with. And so you can change the receptor, so you can change the tissue tropism that this virus can bind to. And so we will talk about this briefly in the Viruses in Biotechnology lecture that you'll look at soon. Okay, I hope that helps. Thanks.